The purpose of this video is to give you a guide through some of the more technical aspects of Project 3. It's a little difficult sometimes in text to troubleshoot all of the various little glitches or obstacles in students' way. So you can follow along and pause the video if needed. Additionally, if you have any trouble or any problems that you're unable to resolve by watching the video, please feel free to contact your instructor and he can help you with those. So we're going to begin by downloading the transcriptome file that's in the project folder. So if you go to Blackboard, in the project folder, you'll see this file that says transcriptome assembly file. Now, it's a rather large file. It's just a text document, but it contains many potential transcripts, that is, gene sequences from a mystery organism. This is a transcriptome that I had sequenced a few years ago, so we're going to use it as an example. So when you click on that, it will download it. Again, it might take a while depending on the speed of your internet connection. So once you've found that, it will be in your downloads. Once you've downloaded that, in your download folder, you'll have that transcriptome. So go ahead and click on that and open it up. Again, it's a large file, so whatever your default text editor is, if you've installed Mega, it might try to open it in Mega because it recognizes it as DNA. But so if clicking on it doesn't work or you're having problems with that, the best thing to do is open your text editor. Now, in the project guide, I've suggested a text editor that you can download for free if you don't have one on your computer. There's one that comes with Microsoft. Apple has one. Sometimes, again, because of the text size, those don't work really well. So if that's an issue, just do a quick download and install of this text editor. It's a very handy little text editor. So once you have opened it up, and you can do that by File, Open, and then navigating, or just by dragging it into this screen. I want you to do a quick observation. So what you will see is that you have, for every potential transcript, a name, and the names are assigned with just a code. So basically it's kind of sequentially names them from this assembly. The next little bit is a value that represents how long in terms of nucleotides your data is. So this transcript here is 454 sequence or base pairs long. The last part is just kind of how many different assemblies, how many different individual small reads during the transcriptome sequencing process were put together. So that's just something the computer uses. We're not going to pay attention to it. The main thing we're going to be looking for is this length. So notice as we scan through here that lengths vary widely. There's some little short pieces that might represent a part of a gene or maybe some artifact of the sequencing or assembly process. So what we're looking for are the larger ones, which are much more likely to represent a gene, maybe an entire gene or a very large portion of it. So if we look on our project guide here, it says, look at it. What do you notice about the length of each transcript? So just need to put something here about length varies, right? Some of them are long and some are much shorter, right? And realize that genes, even if we had every single complete sequence for every gene in this sample, we would still have quite a bit of length variation. Some genes are just bigger, some are shorter, but invariably, because of this kind of shotgun approach where you're going to find everything you can and then evaluate it afterwards, there are going to be some that just don't represent entire genes or some that are artifacts. So your next step is to choose a transcript that's longer than 1,500 base pairs. The main idea here is to find a gene, maybe not a full gene, but probably close to or potentially a complete gene. So I just scan through it, and again, it's a huge file. So notice as I click this, the little indicator of what part of the file you're on just barely moves. There's something like 20,000 or more transcripts in this file from this project. So here's one. Let's just start with this one. So the length is 2,152 base pairs. That seems pretty reasonable. So I'm going to highlight the name and the sequence for this potential gene. You can right click on it and copy or just use Control C, but we're going to copy this, and then next is to do a BLAST search. Right? So if you follow along, it says select one, highlight it, and then perform a BLAST search. BLASTX is probably your best option here. Now BLASTX translates it to an amino acid sequence first before sequencing it. So you have a little bit of a more conserved starting point because amino acid sequences tend to be a little bit more conserved. So let's do that. So I'm just going to go to BLAST, and here's the BLASTX option. So we'll try that, and I'm going to paste it, Control V, or right click paste. Paste now my sequence into this window. Okay, we're now going to perform a BLAST search, and this might take a couple minutes depending on the length of the sequence. It's got to search the entire online database. And what it's doing is looking for the closest matches that are in that database. BLAST is an algorithm that runs through a process based on sequence similarity or the homology of other online sequences. And so it will give us a list of results that shows us the closest match, what is the best score, and then a list of other matches that are similar to our searched gene. Now, if this occurs, it means that there wasn't a close match for that gene. Now that's unusual, but it might happen. I just picked this one at random. So let's try another one. Now, I'm going to give you a alternative one once we find one. I don't want you to do this over and over again, which is unlikely because it's a small number of genes that are like this. But let's do this. I'm just going to scan down and find another one. So here's another gene, 2300 base pairs long. Let's try this one. We're going to do the same thing. Copy, come back to my BLAST window, and we'll paste that one in instead. So here's our new one. Again, we're searching under BLASTX, which translates it into a protein first and then searches based on that protein sequence. Okay, so now we have results from our search. So notice the very first one is listed with a name, and then we have a whole bunch of others. And often what you'll see is notice that we get the same name over and over again, but from different species. That's a really good sign. The other thing we like to look at is this E-value. Now, depending on how variable this group of proteins is, we might have an E-value. This is 3 times 10 to the minus 20th. The smaller this number is, the closer it is to an exact match. So that's a good sign when this has a minus 20 or minus 50 or anywhere above that. I guess I should say below that because it's a 1 over, right? But any small value there is a very good sign. So it looks like we found a good match for this. So the next step, based on the results of our BLAST search, what's the most likely organism from which this transcript is derived? Now notice that our matches here list the taxon, the species from which they came. Now if you don't recognize these, which is very likely don't, you can click on one of these and then it will tell you what species it's from. So notice Ephemera danica, it gives you a lineage. And if we scan through this, we see, oh, it's a metazoan, an animal, right? It's an arthropod. And then we have all the subcategories. And notice that if we go down a little bit farther, we get a list of them. And in fact, often then it will give you the common name also. So this is a mayfly, which is the order Ephemeroptera, an insect. Okay, so you can be very, very specific. The farther you go down this list, the more specific we're getting. So this is the genus all the way down, and we had the family name, or the species name also, Danica. But this is just the closest match. So it's probably not in the same species because it's not an exact match. It's maybe not in the same genus, but it's probably somewhere in this category. Now, in fact, I know ahead of time, this is actually a different member of the family. Ephemeridae was the organism from which this transcript was derived. So just if you're curious, this is the species. It can be found locally. This one, the sample was collected from Central Texas. It was an extraction of the head tissue and then sequencing of all of the mRNAs, the transcripts that were found in that organism. Okay, so again, you can get some indication based on those sequence similarities. So we'll come in here and say, probably our best answer is, the closest matched was a mayfly. And if you wanted to, you could say, order Ephemeroptera. So it's likely the sequence is from a closely related species. Now again, I had some inside information. I know where it came from, but you can get a good indication based on these sequence similarity searches. Okay, so done with number 5. The next step, select the first match from our BLAST search, navigate to the amino acid sequence for this match, then copy the sequence and paste it into a search window at this website. Now this website, Interpro, is a website that looks for structure in your amino acid sequence, looks for matches amongst all of the protein databases, and then tries to assign your protein to a particular family of proteins. So we talked about families of proteins and how they have common ancestry and sequence similarity, and that's what allows us to classify them. And they're often going to have a kind of a closely related function with some variability among that family. So we're going to go to that site and then perform a search. This is one of the reasons I have the video, so it can be sometimes a little difficult to describe how to navigate through all of this. So I'm going to come back to my original BLAST search. I'm going to click on this first match. So here, hypothetical protein, and the names can vary widely depending on how they were found. If it was a genome sequencing project, you'll see a little information about how it matches your sequence, which is on the top line, how it matches your target sequence. But we're going to go to this first match. I'm going to click on this link, which is the sequence identification link. And it's going to take me to a page with all the information about this protein sequence. And at the bottom, we've got a sequence for it. Now, this format can be a little bit, it's not the most common format. So I would recommend you click on this link, which changes it just to a simpler format. A FASTA format, just like in your transcriptome, has a name of a sequence first with a little greater than sign at the beginning. And then the sequence follows it on the next line. So we're going to highlight this. We can do both the name and the sequence. And then we're going to go to that website that's listed. We're going to click on the search by sequence link here in the bottom. And then we're going to paste our sequence in FASTA format. Okay. Once we've got it there, we're going to do the same thing. This is going to take, again, a little bit of time. It's going to search for a match for this protein. If it's similar to other proteins, if it can be classified into a protein family. Now what we'll find is that the vast majority of proteins can be classified based on their similarity to other proteins into a family. And that group of proteins, there may be several genes that are members of that family. In the larger ones, there are even thousands of different genes that are part of that larger family of genes. So we just picked one at random, so we could have very different results here. So your results may vary, but you should end up with very, very good chance, high chance. Now, if your search completes and you don't have it classified as part of a family, very unlikely, but if that happens, I will give you a link to another, I'll give you another target protein for you to work with. I'll help you navigate to one in the transcriptome. So we'll do that here in just a little bit. Okay, so our search is completed. We can click on the results here for this, and it will give us a report about this protein. So this one, in the case, it doesn't seem to be similar enough to other proteins. Now, So no single like small one, but it does have some similarities to others. So it has this very similar superfamily, right? It has these structures that define this superfamily. So although it doesn't have a single family, it does have some, maybe not um, very, very close similarities, but some other ones. So let's just choose one of these and we can complete the project. Now, if it says no matches at all with no similarities, very, very unlikely. You might have a little bit of an issue doing this. But let's select this larger superfamily of proteins. Oh, sorry, here it is over here. Okay, and it's going to give us a list, and it gives us a description. It gives us some references. It will then look at, some of the subfamilies and other things within these. So I'm just going to pick one of the subfamilies for this. So you will have a description of this gene, and then at the bottom you will have a GO term. Now GO is an acronym. It stands for gene ontology. So what I'd like you to do is just read this. They're usually quite short. It'll tell you kind of what happens. So this is a related, maybe distantly related one that has a specific function. Okay. So you may very well have found a different closely related family of proteins. I'm going to give you another potential one. I'll see if I can paste a link to this. I think that I should be able to give you a specific link. So the first one that I did when I was going through this process, we'll go back to our search window here, was this protein. Okay, so your results may look like this. So this one is identified as part of this aftaphyllin-like protein family. So if we click on that protein family, it gives us a description. So this family of proteins, which we found in our transcriptome, represents a group of related proteins from animals. Okay, so it kind of gives us a taxonomic group that it's found in, and it is, it's given names and it tells what it does. It forms a stable complex with other proteins, and then it plays a role in membrane trafficking, right? So moving proteins from one part of the membrane to another, or from one cell membrane to another cell membrane. And it also gives us something a little bit about its secondary structure and any motifs, right, that might be found in that. Okay. So paste the summary description of your identified proteins in this below. So here we go. Here's our summary description. We're just going to copy this and paste it here. Okay. Now, based on that gene ontology, what biological processes does this family of proteins perform? And sometimes you'll have more than one. There might be a pleiotropic gene that does a couple of different things, but for this one, we just have a single one. So it's listed as intracellular transport. I can just copy and paste or type that into here as our answer for this. Let's just do this. I'm just going to paste it in so it doesn't format it. So there we go. It's an intracellular transport. Now the last thing we're supposed to do is, what is the taxonomic range for this family of proteins? Now sometimes your description will do that. So it says it's found in animals, so that would be a taxonomic range. You could say metazoa, the technical term for animals. But notice also over here we have a number of links. It tells us about the different proteins in this family and about the taxonomy. So if I click on that taxonomy link, it's going to give me an overview. Okay, so how many sequences are found in this? Basically, that tells us the different ones. So notice that although it said metazoa, it's in the eukaryote, and every single member of this protein family has been found in an animal. Not only that, but if we look at it, the majority of them are from chordates, with some from arthropods, some from nematodes, and some from mollusks, and then a bunch of other like really small rotifers and cnidaria. So these are all the other animals. So it looks like it's been found across many, many different genomes, but it's an animal gene. So this is one of those genes that defines animals as a group. Okay. So again, your protein might be different. I'd be interested to see what we find. If you run into a dead end or a stop, try again, just pick a different protein and start over. I don't want you to do it over and over and over again. So if you've tried twice and are having problems with it, feel free to use this one as your example protein. Okay, so what I'll do is paste a result for this specific protein that I found. I'll make a link to this. So I can copy the URL for the results for this, and I will post that there as the backup protein, we'll call it. It will be in the folder also. Okay, so once we get done with this, we'll put our last answer here. What's the taxonomic range for this protein? Found, oops, let's do this. It just helps me to see it a little better. I've changed the font color to a different color here just so I can see your answers a little better. If you don't keep it that way, that's okay. But this is found in all animals. Right? So when you get done with this, what I'd like you to do is go ahead and attach this to the project assignment. Okay, so I will put this file in the folder itself. I'll also put it as an attachment to the Project 3 assignment. Just go ahead, open up Project 3 assignment, attach this saved file with your answers in it, and I will review those again. If you have any trouble at all, any technical glitches, or you're trying to do something and can't figure out what you've done wrong, please feel free to send me an email, contact me. I can walk you through it rather quickly if you do run into those sorts of problems.